Hi friends, I am Rahila. My seminar topic is Class Ascidiaceae. Ascidiacea is the largest group of urochordates. It comprises mostly sedentary and few free swimming tunicates, which may be simple or compound and solitary or colonial. Salient features are absence of notochord and post-anal tile in the adult, prominent oral and atrial siphons, spacious atrium and dorsal atriopore, large and well-developed branchial sac with transverse rows of ciliated gill slits, looped intestine and absence of specialized digestive glands. Non-chambered and non-valvular heart enclosed within a thick pericardial tube. Degenerate nervous system in the adult represented by a solid nerve ganglion and a few nerves from it. Absence of specialized sense organ in the adult. In colonial forms, zooids may be either simple and separate with individual testes or composite with a common test, common cloaca and a common atrial siphon. Examples are Ascidia, Herdmania, Siona, Clavellina. Ascidia, sea squid. Ascidia, popularly known as sea squid. It is a sedentary marine chordate remaining attached to rock and other substrata in large groups. Its body is roughly oblong, almost cylindrical and 7 to 10 cm long with a broad base for attachment. At the free terminus is a wide aperture variously known as oral aperture, branchial aperture or mouth. A little behind the mouth and located on one side is the atrial aperture. Branchial aperture is situated on the summit of small prominence called branchial siphon or oral siphon, and atrial aperture on the summit of atrial siphon or excellent siphon. The main body of Ascidia is encased by a protective integument known as test or tunicin. Test is protective, anchoring, respiratory and sensory or receptor in function. Test is unique among animal tissues in that it consists largely of a ground substance called tunicin. Tunicin is a complex polysaccharide closely related to cellulose. The presence of tunicin is a unique feature of ascidians. Tunicin is believed to be similar to mammalian connective tissue on the ground that mammalian dermis contains some fibers having cellulose. The body wall underlying the test is called mantle. Mantle is contractile. By its sudden contraction, jets of water can be forced out through the atrial aperture. The name sea squid is derived from this feature. Digestive system U-shaped with a mouth and anus. Pharynx, largest organ in the body. Food collecting apparatus. Circulatory system shows periodic reversal of blood flow. Their head can contract in opposite directions alternatively in every 3 or 4 minutes. Nephrocytes help in excretion. Ascidians are oviparous hermaphrodites. Their reproductive system consists of an ovary and a testis. Fertilization is generally external in the seawater. Cross fertilization is the general rule. Each fertilized egg undergo embryonic development in about 8 to 24 hours. A free swimming larvae hatches out. It is called a Ascidian tadpole because of its general resemblance to amphibian tadpole. Ascidian tadpole. Ascidian tadpole has an oval trunk and long, laterally compressed and vibratile tail. The whole body is covered by a thin tusk. Trunk or the main body of the larvae bears adhesive papillae at its anterior end. The tail of the larvae is fringed with a median flap-like outgrowth of the tusk. It forms a caudal fin. In the longitudinal axis of the tail is the notochord. It extends along the whole length of the tail. The digestive system of Ascidian tadpole is well developed. Alimentary canal consists of the mouth and large pharynx with gill slits and endostyle, short esophagus, bulb-like stomach and looped intestine. Just below the pharynx is the heart enclosed within a tubular pericardium. The nervous system of the larvae consists of a long dorsal and tubular nerve cord. It has four parts, namely cerebral cone, sense vesicle, visceral ganglion, and spinal cord. Retrogressive metamorphosis. After a short free-swimming active life, tadpole larvae undergoes metamorphosis. During the free-swimming life, the larvae is non-feeding, geonegative and photopositive. So it swims on the surface water in daylight. Later, it becomes geopositive and photonegative and swims downward in darkness. Soon, it settles down to the bottom and attaches itself to a suitable substratum in an upside down posture with the help of its adhesive papillae. Rapid metamorphosis follow. Selection of the substrate is believed to be accomplished with the help of photoreceptors, georeceptors, and tactile receptors. The metamorphosis of Ascidian tadpole characteristically involves the degeneration and disappearance of most of its advanced feature. So, metamorphosis is not progressive and it does not involve a transformation from a simple or lowly organized state to a complex or highly organized condition. On the other hand, it is a radical reversion from a highly organized to a poorly organized condition. So it is called retrogressive metamorphosis. The retrogressive metamorphosis of Ascidian tadpole involved the following major changes. Regression and disappearance of the tail associated with the disintegration and dissolution of the notochord, caudal portion of the nerve cord, tail muscles, etc. Disappearance of sense vesicle, ocellus and otocyst. Reduction of the visceral ganglion and its transformation to the nerve ganglion and the subneural gland. Some workers hold the view that visceral ganglion completely disappear and so nerve ganglion and subneural gland are neoformations. Specialization of the alimentary canal with the enlargement of the branchial sac and the multiplication of gill slits. Development of heart, blood vessels, gonads and gonoducts. Formation of an encasement of testor tunic. Invagination of the test to form branchial and atrial siphons. Replacement of adhesive papillae by ectodermal ampullae.